Okay, a polypeptide molecule is a chain of amino acids. A protein consists of one or more polypeptides. A protein shape is described by 3 or 4 levels of structure. The primary structure of a protein is the sequence of amino acids in each polypeptide. 2. The secondary structure of a protein is the local folding patterns within short segments of each polypeptide due to hydrogen bonding, weak chemical bonds. 3. The tertiary structure is the folding patterns that result from interactions between amino acid side chains, parts of an amino acid, in each polypeptide. These folding patterns generally occur across greater distances than those associated with the secondary structure. 4. The quaternary structure is the result of the clustering between more than one folded polypeptide. A protein can adopt different shapes, and each shape has a relative energy. Lower energy shapes are more stable than higher energy shapes, and a protein with a relatively high energy shape may denature or unfold, and then renature, refold, adopting a more stable shape. A protein that is almost completely denatured is called a random coil. Random coils are unstable because they are high energy shapes. However, some can renature, adopting more stable shapes. 2 scientists discuss protein shape. Okay, so I picked this particular passage because it has a lot of information that most of you probably aren't totally aware of. Even if you know what polypeptide chains are, etc., you might not know what quaternary structure refers to. But it doesn't matter because, like I said in the intro video, this isn't a science knowledge test. It's a ability to read scientific data and comprehend and interpret that information test. So it teaches us everything that we need to know, and all of the things that we need to know to understand what's going on are held within this intro. And P.S., this is a little more extensive intro than a lot of the passages give you. They're normally just, one paragraph, about the size of the last one, that starts with a protein can adopt different shapes. So we've got some key pieces of information. First off, we're discussing protein shape. So, we'd want to underline that. So this is all going to be about protein shape. There's, depending on, what parts of this you're familiar with and what parts you're not, there's a couple different things you could underline. One of the biggest things that seems important is that lower energy shapes are more stable than higher energy shapes. So I would underline that, but I'm going to go ahead and write low energy equals stable. So low energy is stable, or more so than high energy. And it also says a protein it denatures and then renatures. So it has to denature to renature. That seems important. So denature to renature. And that's all talking about to, low energy or stable. Okay, and then in terms of the first 4 things, it explains everything. So if it makes it easier for you, you can underline a couple words within each one of those. But the idea is like, okay, primary structure, sequence. So I could say, primary structure, and I would underline sequence. Again, I know I'm writing stuff on the screen, but that's not what you would be doing. You'd be underlining it. It's just, I don't have the passage on the screen, so I can't just underline things. So primary structure is sequence. Then it gives you secondary structure, which I'm naming 2, structure. So we're looking at like folding patterns, and that's due to hydrogen bonding. So let's say that's H bonds, folding. Notice I'm only going to underline a couple things in each little section. Tertiary or 3 structure, that's between amino acid side chains. So this is like, bigger folding basically. So this is still folding related. This is side chains. And then quaternary or 4 structure, that's clustering of more than one polypeptide. So I'm going to say multiple. You could write multiple next to polypeptide if you needed to. Polypeptides. So I'm just going to call it a polypeptide. Okay, so here is all the stuff that I would be underlining. It's really not that much. Okay, and then pursuant to our little 5-step method, I'm going to also read Scientist 1. So Scientist 1 says, the active shape, the biologically functional shape of a protein is always identical to the protein's lowest energy shape. Any other shape would be unstable. Because a protein's lowest energy shape is determined by its primary structure, its active shape is determined by its primary structure. Okay, so Scientist 1, I would want to underline the active shape is the protein's lowest energy shape. So active shape equals lowest energy. And then also I would just underline is determined by primary structure. Okay, so we've got our little pieces of information. Now, instead of reading Scientist 2, like most people kind of naturally want to do, we're going to go ahead and scour our questions for anything that we can use right now. So 29, I don't see 1. 30, I don't see 1. 31, Scientist 2. 32, don't see 1. 33, which of the following? That's both of them. 34, I see Scientist 2 in there, and 35, I see Scientist 2 in there. Okay, so the reason that that's happening for this one is that a lot of the questions we didn't see Scientist 1 or 2 in, aka 29, 30, and 32 all seem to not mention the scientists. What that means is because this particular passage has such a large intro, they're going to ask you more questions on it. So 29, 30, and 32 all seem to be intro kinds of questions. So we could try to answer them at this point, but we don't actually know if for sure they won't also reference information from Scientist 2. So I know this is kind of a little bit weird of a passage. Normally you'd have at least one Scientist 1 question. So we're going to go ahead, and since we looked for Scientist 1, we will now read Scientist 2, and then we're going to go through, look at Scientist 2, and then everything else. Okay, so Scientist 2 says, the active shape of a protein is dependent upon its primary structure. However, a protein's active shape may also depend on its process of synthesis, the order in time in which the amino acids were bonded together. As synthesis occurs, stable local structures from within short segments of the polypeptide chain due to hydrogen bonding. As synthesis occurs, stable local structures form within short segments of the polypeptide chain due to hydrogen bonding. These local structures may be different than the local structures associated with the protein's lowest energy shape. After synthesis, these structures persist, trapping the protein in an active shape that has more energy than its lowest energy shape. Okay, so off the bat, the first sentence is something that they agree on. So that's pretty cool. So I might write down agree next to that first sentence. So by saying that the active shape is dependent on primary structure, that's active shape equals lowest energy, which is dependent on primary structure, is what we wrote in our little map for Scientist 1. But he has a different idea as well, which is that it's also dependent on the process of synthesis. So the process of synthesis is also important. So if pieces of the polypeptide chain were already in another shape, then that's going to affect, our active shape. Okay, so we can have an active shape that has more energy. So active can have more energy than lowest. Okay, cool. So let's go ahead and look at the questions and see if we can see a scientist too. So we already said that 29 and 30 are kind of both or overall. 31 says both. 32 doesn't mention either, so it's a both or an intro. 33 is both. 34 is both. 35 is a scientist too. So that's going to be the first one that we're going to do. And I'm actually going to go ahead and erase this page so that I can write just the questions and we can work on those. Okay, so if anybody hasn't written down all the notes, go ahead, pause the video, and then we'll work through it from there. Okay, so looking at 35, it says Scientist 2 says that a protein may be trapped in a moderately high energy shape. Which of the following findings, if true, could be used to counter this argument? Okay, so this is just like a not question. So, it says counter, and it's just like not. You might want to underline it so that you make sure you're totally aware that we're kind of dealing with double negatives. So, since we're looking for which of the following findings could be used to counter the argument, we want to find something that says a protein can't be trapped in a moderately high energy shape. So we want proteins to not be able to be trapped in high energy shapes. Okay, A. Once a protein has achieved its tertiary structure, all of the folding patterns at the local level are stable. Okay, so if, once it's at its tertiary structure, everything is stable, that's actually totally fine. Scientists too would agree with that. Because the idea is that, it has, when it's in its primary structure, that's when, the difference kind of happens. That's when it can get stuck in a higher energy state. But once it's fully formed, and it's okay. It's just, in a higher energy state than it normally would be. So A actually kind of agrees, it definitely agrees with Scientist 2's findings. So we have B. Enough energy is available in the environment to overcome local energy barriers, driving the protein to its lowest energy shape. Alright, so if there was enough energy available that was going to overcome the local energy barriers, which again, that's the reason why proteins are able to be stuck in a higher energy level, then it wouldn't agree with the scientists, because if the protein returns to its lowest energy shape, that's contrary to what Scientist 2 is saying. So already I had picked B, but let's check everything. So C, during protein synthesis, the secondary structure of a protein is determined before the tertiary structure is formed. Yep, that's completely true, and that's from the intro actually. And remember, the intro, both of them completely agree with it. The intro is infallible. D, proteins that lose their tertiary structure or quaternary structure also tend to lose their biological functions. Alright, so that does not counter our argument at all, and it's kind of out of scope for what Scientist 2 is talking about. It's true, but it's a little bit random in the context of this passage. So B would definitely be the correct answer. And on the test, by the way, be careful when you're doing conflicting viewpoints not to just start filling out from the first question. Fill out the last one if that's the first one that you're doing. A good way to do that is just fill out on the actual piece of paper all 5 to 7 of your questions, unless you're running out of time, and then go back and put it on your Scantron, because unfortunately it's a really easy mistake to make, and if you start at 29 instead of 35, then you could easily lose, all 7 of these questions, all the points for these questions, so you don't want to do that. Alright, so perfect. We dealt with that. Now let's go ahead and look through the rest of it. So we're going to go ahead and look at all of our both questions. So let's go ahead and start at 29. According to the passage, protein shapes with relatively low energy tend to... Okay, so this is from the first part. It's in Scientist 1, it's in Scientist 2. So proteins that are at a low energy shape are stable, right? We wrote that down / you underlined it in your passage. So they tend to be stable, would be a really good prediction based on what we have written down. All right, so B, random coils. No. Lack of primary structure. Well, that doesn't make sense. They need a primary structure. Otherwise, you don't really have a protein. Become denatured. No. That's only, they'll become denatured and then renature into a lower energy level. Maintain their shape. Yep, maintain their shape. Sounds pretty stable to me. So D is our good answer. Okay, next one. The information in the passage indicates that when a protein is completely denatured, it still retains its original. Okay, and then it's asking about the structures. So if we're looking at the structures, we're just trying to figure out what would happen if it loses its shape. So 4 is about folding, which is shape. 3, what we mean by 4 is quaternary structure. Tertiary structure, again, is folding, that shape. And this is all just based on the stuff that we underlined, by the way, so you don't have to go back and reread anything. You don't want to waste your time doing that because you don't have very much time for the passage overall. Secondary structure is still folding patterns, so it's not that. Primary structure is just the sequence of amino acids. So no matter what the physical structure is, the amino acids are going to be the same. So our primary structure is just going to be the same no matter what shape, the rest of it is in. So F is the right answer there. Okay, so let's go ahead and look at 31. Scientist 2's views differ from Scientist 1's views in that only Scientist 2 believes that a protein's active shape is partially determined by its. All right, so we underlined this as well. Notice the kinds of things that you underline are going to be the kinds of things that most of the questions are on, if not all of the questions are on. So it makes your life a lot easier if you've already done some notes ahead of time. So we know that Scientist 2 agrees that active shape is dependent on primary structure, but we said the process of synthesis is partially responsible for the overall active shape of said protein. So we're looking for the process of synthesis, which, ding, 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 is C. So C is the right answer there. In terms of the other things that it says, quaternary structure, tertiary folding patterns, Scientist 1 and 2 don't actually talk about that. And then amino acid sequence. Two proteins are not the same unless they have the same amino acids. So if a protein's active shape is dependent on its amino acid sequence, that's saying that a protein's active shape is dependent on being a different protein, basically. So it doesn't quite work out. All right, so next one is question number 32. A student has 100 balls. The balls are various colors. The student chooses 15 balls and aligns them in a row. The spatial order in which the balls were placed corresponds to which of the following levels of structure in a protein? Okay, so this is a little bit of a strange question, but basically what they're doing is they're using an outside visual representation and asking you to match it. So it's kind of the opposite of what you normally have to do, where you have to make the you have to make the visual representation based on the passage. It's giving you one in words, but it's still giving you one. And then it's asking you to translate that back into the passage. So the idea is that you have different colors of balls. So I'm just going to randomly say, we have blue, yellow, red, then another red, then a blue, a green, a pink, a red. Let's see, 1, 2, 3, 4, 5, 6, 7, 8. 2 blues to get up to 10. A yellow, a yellow, a yellow, a pink, and a green. So to get us to 15. So this is just a, a visual representation based on what they told us. So we have 15 balls. There are some repeating. There don't have to be, actually. It just says various colors. So they can be whatever colors you want them to be. But I have, some of them are repeating, some of them are not. So the idea is that these are just set out in order, and we're just dealing with different building blocks or different little pieces that are put in a row. So if we're talking about structure, these being the pieces that build up our total row of balls, we're basically dealing with little amino acids, the sequence of them, and they are making up a protein. So this would be just your first or your primary structure, because it's just dealing with the building blocks that you have inherent in the protein rather than the relationship between them or the attraction between them in the folding. So primary structure is what we want. So that would be F. Okay. So next up we have 33. Suppose proteins are almost completely denatured and then allowed to renature in a way that allows them to have their lowest energy shapes. Which of the following statements about the proteins is most consistent with the information presented in the passage? Okay. So the idea is that if we have proteins and they denature and then renature so that they have their lowest energy shape, when we're talking about lowest energy shape, we've also been talking about active shape. So the answers are probably going to have to do with active shapes. So we know that scientist 1 says that the active shape is always identical to the lowest energy shape. And scientist 2 says that the active shape can have more energy than its lowest energy shape. So we've got two kind of slightly differing opinions on what an active shape is versus a lowest energy shape. So we have all lowest energy shapes. Then let's look at the answer choices. A, if scientist 1 is correct, all of the proteins will have their active shapes. Well, that's true because scientist 1 believes that the active shape and the lowest energy shape are synonymous with one another. They're the same exact thing. Okay, so we like that. That's a good answer, but let's, as always, we're going to look at all of the answers. P.S. as a side note, if you're taking this on the real test, you don't have to do what we do and go through all the answers. I just do so that if there are differing opinions on what the right answer was, you can get a little explanation as to why there's only one that actually works out. But on the real test, you don't have time to read every one and explain to yourself if you've already made a prediction, matched it, and it's a good match. Okay, B, if scientist 1 is correct, all the proteins will have shapes different than their active shape. Okay, that's exactly opposite of what we predicted and exactly opposite of what scientist 1 says. C, if scientist 2 is correct, all the proteins will have their active shapes. Not necessarily, because some proteins have an active shape that is not the same as their lowest energy shape, according to scientist 2. And then D, if scientist 2 is correct, all of the proteins will have shapes different than their active shapes. Again, not necessarily true, because active shape of a protein is dependent on primary structure, and some proteins have an active shape that is the same as the lowest energy shape. Scientist 2 just says that not all of them do. So definitely A is the right answer here. Okay. So let's go ahead and look at 34, which we already can see is a visual representation question, although this one is different than 32 in that 32 gave you a picture, or gave you an idea that you can make into your own picture, and then you're explaining it, versus 34, you're modeling it and then matching your model with one of the answer choices and seeing which one is the best match. Okay, so in 34, which of the following diagrams showing the relationship between a given protein shape and its relative energy is consistent with scientist 2's assertions about the energy of proteins, but is not consistent with scientist 1's assertion about the energy of proteins? Okay, And if we just glance, we have relative energy, and then we have our different shapes. So we know for sure that the difference between scientist 1 and scientist 2 is the active shape versus lowest energy shape idea. So we want to see something that, let's see, we want to see something for sure the relationship needs to be, if it's not going to be consistent with scientist 1, is we want to have our low energy shape be lower than our active shape. So knowing that it's going to talk about the lowest energy shape and active shape, we already have a good idea of what's going on. And then if we go ahead and glance at the answers, we actually have a third randomly coiled shape, third category that we didn't write down in our prediction. But we actually, even without that, let's see if we can get it, and then if not, we'll think about what the randomly coiled shape should be relative to the other two. So if we look at F, we have randomly coiled shape, and then active and most stable are the same. Okay, that doesn't match either of the scientists because the lowest energy shape should be your most stable shape. Okay, so then H, again, your active shape and your most stable shape are the same, and that's a problem because we want them to be different in order to not agree with scientist 1 but agree with scientist 2. Okay, in G, they're different finally. That's good. So we have randomly coiled shape, then the active shape, then the most stable shape. So the most stable shape is higher energy than the active shape, which is the opposite relationship than what we drew and what we want. So we are left with J, which has the randomly coiled shape is the highest energy relative, then the active shape, then the most stable shape. That matches what we drew. So the answer is J. And also, if you're wondering about randomly coiled shape, they do mention randomly coiled shapes, and the idea is that the first passage, it says a protein that is almost completely denatured is called a random coil. Random coils are unstable because they're high energy shapes. So we know that it should be a high energy shape versus active shapes and low energy shapes, which, as the name at least of the second one suggests, should be lower in energy. So it makes sense that we reference that material. So notice a lot of the questions that deal with both also heavily reference and rely on the first intro passage for you to kind of figure out what's going on. And if you ever don't understand a term, a lot of times they are in the first part, and they're nice enough a lot of times to also put them in italics. So a lot of the scientific terms in this section, not in the others, will be in italics for you so that you can easily reference them and then try to figure out what they're talking about. Okay, everybody, great job. That's it for the science section. So just remember, on conflicting viewpoints, you want to be underlining enough information that you understand what's going on in the passage, and remember to try to go through and do passage, the intro, and scientist or person 1, then person 1's questions, then you do scientist 2, and then scientist 2's questions, and finally the questions that have to do with both people. If you remember to always do it in that way, in that order, then you have a much easier time avoiding traps. So, always make sure that you go through and you're systematic. But be careful about what you do when you're filling in your Scantron. Okay, guys, really, really good job, and hope to see you at the next video. Okay, thanks, guys.